Hi friends, welcome to the online lecture in DNA replication of elongation in prokaryotes. Okay, so today I will describe about the elongation of DNA replication in prokaryotes. In the first video series of the DNA replication in prokaryotes, I have described about the initiation of DNA replication in prokaryotes. So you have know that in the first lecture, DNA replication occurs in three steps. That is the initiation, elongation, and the terminations. And in the first video series lecture, you listen about the initiation of DNA replication. So you want to know details about the elongation, I will suggest you that please go back to the JK biology and see the initiation of the DNA replication in prokaryotes, then come back to the elongation of DNA replications. Okay, so today I will be described about the elongation of DNA replication in prokaryotes. First, I have described about the summary again, that this is the circular genome of the coli that contains 2 conjugated sequences. Okay, that is the 4 stages of 9 base pair long, that is called replication recognition sequences, and each contains 4 stages that is found upstream side of the sequences. And so this is the region where enzymes bounds or proteins bounds, and with the help of these formation of replication bubbles occur. There is the another sequences, that is the 3 stages of the sequences found in the genome of the bacteria, and this is the called origin of replication, that is called ORC or ORP, and this becomes 13 base pair longs, that is 80 rich sequences. These 3 stages becomes 80 rich sequences, that is very helpful to formation of the replication bubbles. Okay, so come back to the elongations, that is the formation of the replication fork with the help of the DNA B proteins. Okay, DNA B proteins loaded by the DNA C proteins, and this breaks the hydrogen bonds between the 2 nucleotides. Formation of replication forks occur, and here different enzymes and proteins help in the synthesis of DNA replication. So you will see here that this is the single stranded binding protein, that is the SSB proteins, that maintain single strands of both strands and does not make again re-annealing of the base pairs. So this is the single stranded binding protein that help maintain to the single stranded of the DNA, and this is the DNA B proteins that break the hydrogen bond between the 2 nucleotides. Okay, so this is the DNA G proteins that always attach to the DNA B proteins. When DNA B proteins move, break the hydrogen bonds, new replication force form and again move, then there is synthesis occurs by the help of DNA polymerase third. In case of bacteria, DNA polymerase third polymerize at the nucleotide in 5 to 3 directions. Okay, so DNA polymerase third polymerize 5 to 3 directions, and so again DNA B go to forward direction, break the hydrogen bonds, again new replication forks, and so you can say that for a single DNA replication, more and more replication forks, and at once at the times of the replication fork, DNA polymerase thirds add the nucleotides and synthesize in both directions. So you know that in the elongation only single DNA polymerase third is present, but actually DNA synthesis occur in bacteria in both directions simultaneously. That is the big problems, that is the big puzzles, that DNA polymerase third is only single, and this polymerase synthesize both directions simultaneously. How? Because this is the universal truth, that a DNA polymerase add nucleotides only 5 to 3 directions. It is only made that add nucleotides only in 5 to 3 direction. It does not add 3 to 5 direction. It is not made for 3 to 5 direction adding nucleotides. Okay, so this is the problems for the DNA polymerase. So here you will see that this is the 5 and this is the 3 ends. This is also 3 and this is the 5 ends. That is opposite in directions. So this is the DNA polymerase thirds. That's when primer is attached. DNA polymerase synthesize 5 to 3 direction. But another instance, there is no attachment of primer in the 3 sides. So there is a big problem for DNA polymerase thirds. In this case, first I described that how primer synthesize again, that this is the DNA G proteins, and DNA G proteins synthesize RNA primers, that is the ribonucleotide. This synthesize ribonucleotides, and the ribonucleotides attached to the 3 ends. And RNA polymerase, if this synthesize primers, that is 5 to 3 and again 5 to 3. Okay, synthesized primers. Then these primers is attached by the RNA polymerase to the 3 prime. Okay, so you will point out statements, or you will point that I am saying common questions that may be confused for the students. This is the 3 ends and this is the 5 ends. So RNA primers read DNA strands, template of DNA strand is read in 3 to 5 directions. Point out, this is that DNA template, this is the DNA template. Okay, DNA template is read in 3 to 5 direction, but the synthesis occurs in 5 to 3 directions. This is the confused for the student in the examinations, that examiners can ask that template reading is occur in which direction, that is 5 to 3 or 3 to 5. Then the answer is that template is read in 3 to 5 direction and actual DNA synthesis occur 5 to 3 directions. Okay, so here when the primer is attached in the 3 OH, 3 primes, then the DNA polymerase add nucleotides and move in 5 to 3 direction. But for another instance, there is a problem. There is a DNA polymerase. I will describe first that the structure of the DNA polymerase. That's this is DNA polymerase. Okay, so this is for simple understanding. DNA polymerase solve these problems. That's synthesize both directions simultaneously in case of bacteria by formation of making loops in another instance, that is 5. Okay, makes the loops, and this is the polymerase. Also this is the DNA polymerase third, DNA pol third, and this is also subunit of DNA pol thirds. Okay, thirds. So what would be happens when DNA B proteins break hydrogen bonds, replication forks form. Okay, DNA G synthesized primers, oligonucleotides, RNA polymerase add the primers, but in this case single primer is added here, that is 5 and 3. Single primer added and DNA polymerase recognize and synthesize or add the nucleotides in 5 to 3 directions. But here these moves in this area, but in here you will see that there is a most primer attached. Why? Because the DNA polymerase subunits take the primers. This is the primers again, and it is called the clamp loaders. This is called clamp loader. This clamp loader load the clamps where the primer is attached. Load the clamp. This is the clamp, and these clamps always load where the primer is attached. When the primers become the clamps becomes attached with the primers, then the subunit of the DNA polymerase or arm of the DNA polymerase recognize the clamps and synthesize in 5 to 3 direction. This is the 5 and this is the 3. Okay, so synthesize here, and when synthesized in forward direction, synthesis becomes complete or adding becomes complete, then these become detached. These subunits become detached again and come back. These arms come back and take the another primers where clamps is attached, and catch up those primers and again synthesize in 5 to 3 directions. So again this process will take place by the another subunit of the DNA polymerase and synthesize in 5 to 3 directions. So what would be the results? There is, if it gets primers, then there is no problems in 5 to 3 direction, and always at the nucleotides there is no give chance to attach another primers. At once synthesize or add nucleotides, it takes nucleotides from the cells and add the nucleotides. But here, when these moves till 5 to 3 direction complete synthesis, there is enough time to expose for attachment of the primer. So multiple primers is attached on this strand. So there is the continuous synthesis of the DNA synthesis, and here is discontinuous adding of the nucleotide. That is called the lagging strand. This is called lagging strand. Lagging strands, okay, and another is called leading strands. Okay, so you will see here that there is multiple primers, that is the ribonucleotides. There is no works, there is no need of the RNA primers in the DNA strands. So how it would be the solved? There is another enzymes that remove the RNA primers. Okay, this synthesis, suppose that this was the actual structure of the replication fork and replications where occur, and I will see you again. This is the 5 and 3 here. This is the primers, okay, and the synthesis of DNA polymerase completed in 5 to 3 directions. This is the 5 and this is the 3. This is in case of leading strands, okay, but in another case, that is 3 and this is the 5 primes, okay. Here multiple primer is required to attachments, okay, that is the multiple primers, okay. This is the multiple primers, but it requires removal of the primers, because this is made of ribonucleotides. There is no work of ribonucleotides. So these ribonucleotides is removed by RNase H, okay, or DNA polymerase first, that have 3 to 5 exonuclease activity. What is the 3 to 5 exonuclease activity? Exonuclease activity which removes the terminal nucleotides, not between the nucleotides, not middle portion. That is, here is the T, A, T, that is the here T, and that is the G. This is the terminal, this is the middle portions. This is where this nucleotides is removed, that is called endonucleases. When these enzymes, any enzymes remove that portions of the middle portions of the sequences, that is called endonucleases, and when remove terminal nucleotides, that is called the exonucleases. So DNA polymerase always remove from 3 to 5 direction, that is 3 and that is 5, remove each nucleotide from the terminals, that is called the DNA polymerase first. When the removes, the primers becomes removed, what happens? There is gap, there is many gaps. So these gaps is filled by the DNA polymerase first. Also DNA polymerase first fill these gaps. Point out that DNA polymerase work as both, that is removal of ribonucleotides and adding of deoxyribonucleotides. This is the deoxyribonucleotides, but this not makes phosphodiester bonds between the nucleotides. There is need to attachment, actual attachments from nucleotide to nucleotide need phosphodiester bonds. So it does not make the phosphodiester bonds between the nucleotides. So there is a nick, okay, there is a nick, more nicks, okay, there is more nicks. So this nicks is filled by the DNA ligase. These nicks is filled by the DNA ligase, okay. This was the complete synthesis of the DNA replication in DNA elongations in prokaryotes. But you think that's where energy come from to take the forward direction of the DNA synthesis? How energy come from to complete the process of DNA replication? Again, I will see described on the basis of molecular level, that is this is the nucleotides, okay. this is the nucleotides that contain 3 phosphates. This is the 5 end, this is the 3 ends. Okay, this is the 3 ends. So here if one nucleotides becomes attached, that is the phosphates, this is the phosphates 5 and this is the 3 HO groups. Okay, so this is called alpha phosphates, this is the beta phosphates and this is the gamma phosphates. This is the T, A, G, C. Okay, so these attacks on the alpha phosphate groups, nucleophilic attacks occur, then the removal of 2 phosphates, okay, and adding of these nucleotides from this, and so the nucleotides again becomes as like this. Okay, this is the HO, this is the T and A, G and C. These nucleotides become attached, but pyrophosphate is released. This is the high energy containing phosphates. When these phosphates break, then the energy is released, and which energy, these energy released, that's make the favorable for the DNA replication in forward directions. Okay, so this was the 3 phosphate groups that found in the nucleotides. That's 2 phosphate is released and 1 phosphate attached to the another nucleotide that make the phosphodiester bonds, and the adding of nucleotides occur by the DNA polymerase, and the DNA polymerase makes easy to synthesis of both strands of DNA in prokaryotes simultaneously. So this was the elongation of DNA replication in prokaryotes. Okay, and in the next lecture I will be described about the termination of DNA replication in prokaryotes. Okay, so if you like this lecture, then please hit or click in subscribe button. Your subscription will bring me a big hope to make a good lecture for you. So please click on subscribe button and get a more and more lecture for your competitive examinations. So please help to improve these lectures and click on subscribe button. Thank you. Okay.